When animals and people learn, what changes in the brain? As I said earlier, much of our understanding of the neural correlates of learning comes from the studies on simple systems and simple forms of learning. However, researchers are rapidly identifying the neurophysiological basis of learning in humans as well. Learning involves relatively permanent changes in the brain that result from exposure to environmental events. If we require changes in neural processing, then changes in neural shape, size, number of connections are all candidates for the brain basis of learning. And in fact, all of those have been observed at different levels. Synaptic plasticity, changes at the level of synapses, have been investigated as the basis of learning, and subsequent structural alterations have been seen. Eric Kandel's work on habituation and sensitization in aplysia, sea slugs, showed synaptic level plasticity. Studying classical conditioning, Donald Hebb postulated that connections between neurons increase in efficacy in proportion to the degree of correlation between pre- and postsynaptic activity. One of the most quoted paragraphs in neuroscience is the one I have here. Donald Hebb said that when an axon in cell A is near enough to excite cell B and repeatedly and persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells such that A's efficacy in firing B is increased. Hebb's hypothesis is often paraphrased as Hebb's rule that neurons that fire together wire together, and has in fact been shown to be true at many levels of organization. The idea that repeated exposures can strengthen connections or strengthen networks such that then retrieval of information during episodes of incomplete activation becomes easier has been shown. In many pattern completion tasks, we see priming effects of prior exposure and learning. Complete this word fragment with the first word that comes to mind. The most common responses to this are skirt, skill, skate, skis, and skier, and so on. The less common examples like skimp, skeen, scarn, and skine don't often come to mind. Why is this? Well, primarily because the strengthening of more common words that we are exposed to more, is stronger, and that gets activated first. Some of you may have thought of the word school as thinking of sk as a word stem. This would be priming in the auditory realm. Even though you were looking for sk words, when trying to complete a sk word, school does come to mind, and that has to do with the strength of connections between those sounds. Hebbian learning explains these observations based on the strengthening of connections for more common words, more commonly exposed information, that then comes more readily to mind, or an incomplete pattern of activation is presented. What then is this strengthening? A candidate for the neural basis of synaptic strengthening is long-term potentiation. Long-term potentiation refers to the strengthening of synaptic connections, making the postsynaptic neurons more easily activated with presynaptic input. Through LTP, intense and repeated stimulation of neurons strengthens synapses, increasing the likelihood that one neuron's activation will lead to the firing of other neurons. This strengthening of synapse is long-term in that it outlasts the learning episodes and shows heightened activation in neurons days, weeks, and long after the initial strengthening. LTP effects are most easily observed in brain sites known to be involved in learning and memory, such as the hippocampus. LTP in the amygdala appears to play a role in fear conditioning. Joseph Ledoux's research suggested that fear conditioning showed increased activation in the amygdala when subjects watched another person's distress. This suggests that similar mechanisms are involved in conditioning and observational fear learning as well. How is LTP implemented? LTP occurs when NMDA-type glutamate receptors are activated. Joseph Sines modified genes in mice to make the gene's NMDA receptors more efficient. He called these mutated mice Doogie mice, after the genius teenage doctor in the television show Doogie Howser, MD. These Doogie mice learned novel tasks quicker and showed increased fear conditioning. So then it seems that LTP and synaptic plasticity underlie a lot of learning in the brain. Cortical plasticity, the reorganization of connectivity based on experience and exposures, is an extension of synaptic plasticity at the level of the networks. We've seen several examples of cortical plasticity earlier in the topographies of the visual cortex, the somatosensory cortex. The study depicted here shows a correlation between the extent of activation of the right index finger and the discrimination thresholds for the same index finger compared to the left index finger before and after exposures to a grading discrimination task.
We know a good workout can make you stronger. Now researchers are finding that a brain boot camp can make you smarter.
What we can see after training that people actually got smarter in these tests. So we can actually say that people got smarter after the training.
Suzanne Yaki and Martin Bushkill have been putting volunteers through up to 4 weeks of computerized mental gymnastics. Volunteer Jordan Harris says he's seen a difference.
I used to lose jackets and clothes all the time, and I guess I haven't had that problem recently.
Volunteers took general intelligence tests both before and after the training. Bushkill says the improvement was striking.
They solved 40% more problems, new problems that were presented to them than before the training, which I think is quite impressive.
Writing in the Proceedings of the National Academy of Sciences, the researchers emphasize what's different in their training is that people are showing improvement solving tasks they had not trained for.
In other training programs that are on the market, people get very well in this particular task, but then their ability does not transfer to other domains than the training task itself.
But does 4 weeks of mental gymnastics make you permanently smarter? The researchers doubt it, like physical exercise.
Without pain, no gain.
Which means regular workouts for more than your muscles. I'm Brad Closa.
LTP, however, is not the sole mechanism of learning. Let's take a look at this video, Wiring Better Brains Beyond LTP.
Whether you're starting a new sport or learning in school, your ability to absorb information depends on an expanding web of wiring inside your brain.
The brain is very dynamic. Your brain doesn't just stop growing and doesn't stop making new connections. It actually forms new connections all the time.
Just how this network of nerve cells grows has long boggled our minds, but brain scientist Bonnie Firestein has pinpointed one piece of the puzzle. She and her colleagues studied nerve cells in test tubes. They reported in the journal Nature Neuroscience that a chemical called sipin controls how nerve cells branch out to send and receive signals. The more sipin, the more branches.
So maybe this is what's going on in the brain. When you're learning, you're making the nerve cells active, you're having increases in sipin, and then you're having more branches or more wiring so that you can learn.
That's in a healthy brain. Those afflicted with diseases such as Alzheimer's grow fewer nerve connections.
And the big pie in the sky idea is to somehow be able to develop therapies that would regulate the number of branches and perhaps increase the number of branches and overcome these learning deficits.
But that's years down the road. For now, Firestein will focus on finding what controls the direction of each connection. I'm Brad Closa.
And finally, in understanding the brain basis of learning mechanisms, it's important to talk about the evidence that suggests that dopaminergic activity forms the basis of positive reinforcement, and that release of dopamine serves to increase the desire for a behavior that is rewarded. Reinforcement works in two ways. It provides the subjective experience of pleasure and increases wanting for the object or event that produces the reward. Research over the past 50 years has shown that dopamine plays a very important role in the experience of reward. Evidence for the involvement of dopamine and the pleasure centers, as they are called, came from the work of Olds and Milner, who set up an experiment to see if rats would press a lever to self-administer electrical pulses to specific sites in their brains. These intracranial self-stimulation studies showed that rats pressed the levers hundreds of times and preferred to do so over eating, drinking, and all other activities. This ICSS was shown to activate dopamine receptors in the nucleus accumbens. The nucleus accumbens is a subcortical brain region that is part of the limbic system, the same site in the brain regions as those activated by natural reinforcers such as food, water, and sex. interfering with dopaminergic transmission or lesions to the nucleus accumbens, eliminate self-stimulation, as well as other naturally motivated behaviors such as feeding, drinking, and copulation. Hungry rats that are given food have increased release of dopamine in the nucleus accumbens, and the greater the hunger, the greater the release of dopamine on presentation of food. Think about how much better food tastes when you are hungry, and water is much more rewarding when you are thirsty. This is because of the dopamine release under deprived rather than non-deprived conditions. Even the enjoyable activity of looking at funny cartoons activates the nucleus accumbens. In operant conditioning, the release of dopamine sets the value of a reinforcer. Drugs that block the effects of dopamine disrupt operant conditioning. As you might expect, drugs that enhance the activation of dopamine, such as cocaine and amphetamine, increase the reward value of stimuli and are thus very addictive. This means that both primary and secondary reinforcers rely on dopamine to modulate the wanting for that activity. For the same reason, individuals with Tourette syndrome, a disorder of motor control, who are given dopamine blockers to control their involuntary body movements, often have trouble staying on their drug regimens. This is because they typically report that the drugs rob them of life's enjoyment. So while all processes that underlie learning are not yet clear, we have a pretty comprehensive understanding of some of the basic mechanisms that are involved in learning.
10-year-old Vicky Haskopoulos takes regular violin lessons and practices 3 hours a week. It's long been known this kind of brain-building activity is vital for young children, but brain researcher Jay Geed says it's just as important for teenagers to flex some mental muscle.
The first 5 years are very important, so are the next 5, so are the next 5 after that. The brain doesn't grow smoothly and evenly. It grows in fits and starts, and different parts mature at different ages.
Geed and his colleagues discovered this by creating a unique 3D animation of the maturing brain. The dark coloring indicates regions of development. Using MRI technology, they scan the brains of 13 healthy children and teenagers every 2 years over a 10-year span. The resulting images, published in the Proceedings of the National Academy of Sciences, reveal for the first time the sequence of dynamic brain development between the ages of 5 and 20.
During these times when the brain is undergoing these great changes is the time when practice of a musical instrument or a sport or different studies may have a much greater impact than in earlier or later years.
Just how much greater is yet to be seen, says UCLA neuroscientist Arthur Toga, who oversaw the brain animation. He says as we continue to chart the human brain, that map will continue to be better populated with more information. So it doesn't matter if kids prefer science, sports, or strings. The important thing is that they keep using their heads. I'm Karen Lurie.